Okay, so it's a great pleasure to have Professor Catherine Talon-Baudry with us today. Um, Catherine is a senior CNRS researcher. Initially, she was trained in biology at the École Normale Supérieure in Lyon. She did her PhD work in Lyon, where she revealed the existence of gamma band oscillations in humans and their role in visual cognition. I remember when her PhD came out, it was very influential back then and still is. After her postdoc in Bremen, Germany, she joined the Hôpital Petit-Salpêtrière in Paris in 2002, where she, as she puts it, unexpectedly found a double dissociation between neural correlates of spatial attention and visual consciousness. I really loved it that she used unexpectedly. I think it says a lot about her way of research, very open-minded, very open for discoveries, and very authentic and very deep, asking questions. So I love this exploratory research program myself, following her career. She got intrigued by consciousness, but also dissatisfied, as she puts it again, by cognitive accounts of consciousness. And she thus decided to concentrate on the core property of consciousness, which is subjectivity. In 2012, she moved to École Normale Supérieure in Paris, where she began to work on the link between brain-viscera interactions and subjectivity, finding along the way that such interactions account for a lot of the so-called noise in the human nervous system. It's a real pleasure to have you with us today, Catherine. Thanks for finding the time, and we really look forward to hearing your talk.
Thanks, Moshe, for the kind introduction. And it's nice to see some familiar faces around. Thanks you all for being here. As usual, so I have some people on Zoom. I know that some people are in a seminar room. If ever at some point you have difficulties hearing me, or if my slides don't move, let me know. Don't leave me talking to a blank space if anything unexpected appears. So I'll be talking about interoception and cognition. And as Moshe nicely put it in the beginning, I like big questions. I think this one is one you will like too, Moshe, which is, why do we do anything at all? And this is, of course, a big question, which probably has many answers. So I'll just take a small portion of this big issue. But I think part of the answer is very basically to maintain the organism alive, which means that this basic fact that any organism has to stay alive must be at the root of all goal-oriented behavior. And what I mean by this is that clever goal-oriented behavior must have evolved on top of this basic default functioning of the organism, which is to stay alive. But then it follows that we have probably a very primitive loop, which consists in interrogating physiological states, defining needs, and then act, which probably then change physiological state, and then we get this loop. So this means that this basic fact of interrogating physiological state is probably a driving force, both for evolution and behavior. This is interrogating the physiological state is precisely the definition of interoception. Interoception is a term that was coined by Sherrington at the beginning of the 20th century. And it meant by this visceroception, that is a perception, conscious or unconscious, of activities in the abdominal cavity, like cardiovascular activity, respiratory activity, GI tract activity, bladder activity, etc. And he made the distinction between interoception and proprioception, which is for muscles and joints, and exteroception, which is the perception of the external world. So what is interoception today? Well, that's a tough question. There is no absolute definition. It's rather broader than the definition of Sherrington. In this consensus paper, it was defined as a perception of signals originating from within the body, which is extremely broad, and it could be internal temperature, nutrient sensing, but then should we also include inflammation or microbiota? It's getting really massive. I will use interoception more in the sense of Sherrington, but I have no opinion about what should be the exact definition. And I will be mostly talking about the heart and the stomach. So interoception can sometimes give rise to conscious perception. If you're running after a bus and you're out of shape, probably you can feel your heart pounding, but that's not very usual. The heart is beating at all times, and you will see that there is also some ongoing activity in the stomach, but this is most often unconscious, and this is also what I will concentrate about. There is a bit of a paradox regarding interoception because on the one hand, I think everyone would agree that it's probably something very fundamental for a biological organism. But still, so far, what has mostly been done is about the descending part of this loop, where there is the sensory part, which is interoception, and then there is homeostatic and allostatic control, which is the regulation of internal organs by the brain, and this is what has been mostly studied. If we make the analogy on this interoception regulation loop, there is a clear analogy with the perception, cognition, action loop when it's about perceiving the external world. And then we can see that in this loop, we know a lot about perception of the external world, but we know actually very little of the perception of the internal world. And finally, there is quite a gap between animal studies, which have studied interoception up to the brainstem, very often in anesthetized models, although this is changing recently, and some human studies which concentrated on the cortical part. And I think part of the reason that there is this kind of conception that well, okay, interoception is important, but it's something very primitive, and it's mostly subcortical structures, mostly reflexes. It's not something very interesting from a cognitive point of view. There is this kind of divide where there is what I call the housekeeping functions, of which interoception is part, which is not very interesting for cognitive neuroscientists. And then there is the real cognition, the clever stuff. Which is supported by the cortex, and those are two separate domains, and I hope to convince you that they are not two separate domains. So what I would like to show you is that interrogating physiological state is a default routine which occurs at all times, even in the absence of homeostatic challenge, that it explains part of cortical spontaneous brain dynamics, and that interacts with cognition, and I will make the point that it could be related to subjectivity. So my talk will essentially be divided in those two parts, one which is about spontaneous brain dynamics, and the other one with the interactions with cognition and the link with subjectivity. And I will begin with the spontaneous brain dynamics part. Because I guess you're all familiar with the fact that the heart is beating more or less regularly, but it's beating continuously. There is actually something very similar going on in the stomach. You're probably less familiar with it. I'll come to it in a minute. But essentially, the idea was super simple, which is to think, okay, now we get oscillators, which are in the abdominal cavity. They are autonomous, meaning that, for instance, for a heart, you know that a heart can beat without a brain, doesn't require a brain to beat. And we have a complex system, which is the brain coupled with those oscillators. There must be something in the spontaneous brain dynamics measured at the brain level, which correspond to the activity of those oscillators. So very basic low-level ideas. So I will talk in this part mostly about the stomach, and maybe first introduce you to the gastric slow rhythm, which is not very well known and which I had never heard about until a few years ago, actually. So the stomach is this pouch, and it's composed of two muscle layers, which are orthogonal one to the other. And during digestion, it's the contraction of those muscles which push the food downwards. And in between those two muscle layers, there are very interesting cells, which are called interstitial cells of Caral. And they are not neurons, but they nevertheless have neuron-like properties. So they are intrinsic pacemakers that generate their own electrical rhythm. And those are slow currents in humans at the frequency of 0.05 hertz, which is a rhythm of a period of 20 seconds. And they generate this rhythm at all times, and it's really intrinsic in the sense that if those cells are cultivated in a petri dish, they go on generating this slow rhythm. They are real pacemakers. And during digestion, there is a reflex. The activity of those cells increases, so the rhythm is larger, and then they are coupled with the muscles. And then this generates and orchestrates the contractions in the stomach. But the rhythm is generated at all times. And those cells, interestingly, also have synapse-like contacts with vagal sensory neurons. So potentially, their activity is relayed up to the brain. And in humans, it's actually pretty easy to record the activity of the stomach by placing electrodes over the abdomen, so regular electrodes that you would use for an electrocardiogram. And here is an example of the raw signal one would get. You can see the slow gastric rhythm here. So with a cycle is about 20 seconds. On top of it, you can see the faster respiratory cycles. And then all those vertical marks are the heartbeats, okay, which are zoomed in here. Which means that the stomach has a very distinctive spectral frequency with a sharp peak around 0.05 hertz, meaning that then you know that you get the stomach signal very easily. You don't necessarily get it nicely in everyone, but if you see the signature, then you can be sure that this is a stomach. So we simply ask this basic question, can we consider the stomach as an electrical pacemaker constraining spontaneous brain activity? And we'll first present you results we obtained in fMRI, where we recorded spontaneous brain activity with fMRI and the electrogastrogram with electrophysiology at the same time. And we looked at stomach-ball phase synchrony or delayed connectivity, depending on whether you're an electrophysiologist or someone looking at fMRI dynamics. And essentially the idea that if we have the gastric signal here in blue, we were looking for brain voxels whose time course would follow the time course of the stomach. So being a bit faster when the stomach is faster, being a bit slower when the stomach is a bit slower. And what we found, quite to our surprise, I must say, is a quite large network that you see here in warm colors, where there is more coupling than expected by chance between the stomach and the ball signal. And in this network, the phase of the stomach explained about 15% of BOLD variance. So it's not massive, but it's also large enough that it should be taken into account. And what we also find is that we really get something like what I've schematized here, which is more like a wave, like an organized sequence, where we first get activity in the somatomotor regions, but then the late regions are more in the occipital regions. And this takes me to what was the most surprising aspect of this network, is that we find all sensory and motor regions in this network. And a little bit of the default network here, mostly, as well as here. Which was not at all what we had expected. Honestly, I was expecting a lot in the default network and nothing in sensory regions, so completely wrong. But this is what we get, and this is pretty robust because a similar network has since been observed by another group in rats using fMRI as well. So we also used MEG to investigate this question. And here we looked for phase amplitude coupling. So we looked for brain rhythms, symbolized here in gray, where the amplitude of the rhythm would wax and wanes with the phase of the gastric rhythm. And we found that in the alpha range, there is a strong significant coupling in those regions here, explaining about 8% of the variance of alpha power. So again, not massive, but not negligible either. And the fMRI data, we could not ascertain the directionality of the interactions. So it's just coupling. We don't know if it's the brain driving the stomach or the other way around, or a more complex form of coupling. For the phase amplitude coupling with the alpha rhythm, we could show that it was actually an ascending influence from stomach to brain, so that 8% of alpha power is actually constrained by the phase of the gastric rhythm. So what are the pathways for such interactions? So here it's a bit of a complex slide, but there is actually not so much information in it. If we start from the stomach, so there could be two pathways, one from the vagus nerve, which then projects to the nucleus tractus solitarius and parabrachial nucleus in the brainstem, and to the thalamus, and then to a number of cortical regions, which are not necessarily known, the projections from thalamus. It also projects to all kinds of neuromodulatory nuclei. And there is another pathway through the spinal cord, through the splanchnic nerve and the spinal cord. And so in our data, we have, of course, no idea which pathway is used. In rats, the group of Zhongming Liu could show that if you sever the vagus nerve, it actually reduces a lot the coupling, except in the somatosensory regions, where it's probably then rather due to spinal pathways. So those results were a bit surprising. They said we reviewed the literature. So there is a small literature of people doing gastric stimulation. So typically in humans, it's done by having participants ingesting a balloon, and then the balloon is inflated until people report having, a full stomach sensation or things like that. And in animals, there are all kinds of different techniques which have been used. And if we review this literature, we actually find results which are quite congruent with what we found, with effects notably in the occipital regions, in the visual cortices, in somatomotor regions, but also in auditory regions. So this, and in regions of the default network as well. So this idea that the gastric rhythm is present in all classical perceptual and motor cortices, that we relate more to relationship with the external world, seems to fit with this a bit scattered literature. So what is the functional role of this coupling? Well, I wish I had the answer to that question. The short answer is we don't know yet. And we actually had a couple of experiments that we were super excited about, but that we could not replicate. So we were much less excited about. So at the present moment, we just don't know. So I come to the second part of my talk, which is more the interaction with cognition. And as you can guess, since I've said that we don't know what is the functional role of the coupling of the stomach, that will be more about the heart. But before I dive into the experimental work, I want to go back a little bit to why I started this research line. And it's all related to the notion that to explain conscious experience, we need to understand how we could build a subject of experience from biological material. So what do I mean by experience? Well, in cognitive neuroscience, we very often study skills and abilities, okay? Abilities to do things. From logical reasoning, high-level planning, language, to paying attention exclusively or in parallel, etc., etc. And what is the link between those skills and abilities to consciousness? Well, obviously, we can express our skills and abilities better when we're conscious, but it's not necessarily the most intrinsic part of consciousness. And what is now agreed on is that what is really at the core of consciousness is subjective experiences. And the definition of subjective experiences is just whatever you feel. which can be something very exciting, like base jumping, something very pleasant, like this famous pastry, at least in Paris, by Pierre Hermé, or it can be an aesthetic experience, or it can be just a boring city view. It doesn't have to be fancy or whatever. It's whatever you feel at a particular moment. But this is actually what our conscious mental life is made of. And this is what needs to be explained. And this is, of course, a huge topic in itself. But one thing that I'm really interested in is this notion that subjective experiences require a subject of experience. They don't exist out of nowhere. They are related to a subject of experience. And only if you have a subject of experience does it make sense to talk about subjective experiences. So the question becomes then, how can one build a subject of experience? And there is actually a long tradition suggesting that bodily signals could be important for several reasons. So one is that bodily signals, so they are internal and private. So they are in a way self-specifying, telling the brain that's the organism around the brain. But they are also related to the basic form of self, which is essentially the organism form of self, the organism which needs to be protected, which needs to be fed, which needs to be regulated. And this is thought to be a basis for generating a subject of experience, but there is some magic involved in those three dots. And that's where I would like to get in a bit more into. In this view, consciousness is, of course, generated in the brain, but it's nevertheless a property of the organism. So that in this idea, an isolated brain, like the philosophers love those sort of experiments of an isolated brain in a vat, would it be conscious? In this view, it could not, because consciousness would exist only as a property of the whole organism. So why focus on organs such as the heart or the gastrointestinal tract? Well, they are life-sustaining organs. They are pacemakers, so constantly feeding the brain with inputs from fetal development to death. So there is a certain stability in this, but they nevertheless vary at a time scale which is fast enough for perception and cognition. And so the idea is that the interoception generates a possibility for subjective experience to exist, but it's a subjective experience which can be about the external world. It's not about perceiving consciously those signals. So let me expand a little bit on that. So the idea is that interoceptive signals could help generating some of the properties of subjective experience. One of those properties is the fact that subjective experience is unified. So for instance, if your current experience is to hold the hand of a toddler, then this experience is perceived in skin coordinates because of the touch, but also in gaze coordinates if you are actually looking at the hand contacts. So those two experiences, those two coordinates are separate coordinate systems, and they can be independent one from the other. So you can be looking elsewhere, et cetera, et cetera. You can move your hand. So it means that to generate this unified experience, somehow the brain needs to reconcile those two coordinate systems. This is a very long-standing issue in cognitive neuroscience. And the hypothesis here is that visceral signals, such as from the heart and the stomach, could provide a way to generate this rotation matrix or this integration between different reference frames. I don't have the proof for that. It's just to explain the overall ideas that guided this work. But we've seen, for instance, that the signals from the stomach are actually present in many sensory regions. Okay, so this could actually provide a way to bind those different activities. But that's not the only property that visceral signals could explain. It could also explain the first-person perspective. And by this, I mean that any conscious experience is experienced from a certain point of view, which is related to where you are as a body in space. And it's not only related to where you're looking at. I mean, if you tilt your head like that, I mean, your overall experience doesn't change that much, okay? So it's something which is more related to the body, and there is some experimental evidence to suggest that it's more along the thoracic segments. And finally, another property of subjective experience is what philosophers call intrinsic mindness. So this seems like a very barbaric term, but it simply refers to the fact that whenever you experience something, it's your experience. You don't have to think about it. It's by default, so that's why it's called intrinsic. And it's obvious that it's your experience. It's not the experience of someone else, except in some psychiatric disorders. But here I'm talking about healthy human beings. And because visible signals are private and are signaling the presence of the organism, they could also potentially explain this intrinsic mindness property of conscious experience. Okay, so again, I will not prove that those mechanisms are the one employed. I just have correlational evidence that there might be something true in this hypothesis, but there might also be alternative interpretations. So what kind of experimental work have we been doing? Importantly, in this hypothesis, bodily state does not have to change, because whatever you do, your heart is beating and your stomach is generating a slow rhythm. So you don't have to be, in a very emotional state, or you don't have to do physical efforts. And it does not require that you consciously perceive your visible signals. Okay, it's really more about a mechanism that could allow for the perception of whatever, for instance, of the external world. So to investigate this hypothesis, we looked at the heartbeat evoked response. So I guess here you're all familiar with what a visual evoked response is. Present a visual stimulus, record EEG activity, for instance, so pretty noisy. So you repeat, you get more data, and then when you average, you see a transient, which is a visual evoked response. Well, one can do the same thing, taking the heartbeat as the input stimulus for the brain. So for instance, the R peak of the electrocardiogram, getting neural activity, and by averaging, getting a transient. Okay, so those are simulations. The heartbeat evoked response was first reported in the 1980s by the group of Chandri in Germany, but did not attract a lot of attention at the time. Just to show you an example from our own data from intracranial recordings in the somatosensory cortex in humans, this is time 0 would be the R peak of the electrocardiogram, so this peak, and then you can see this very nice transient about 200 milliseconds later. Okay. So there is a real heartbeat evoked response. For those of you who are familiar with electrophysiology, there are also all kinds of nightmares with artifacts, etc., etc. I won't go into the details here. I'm, of course, willing to answer any questions, but we've thought a lot about this, and we have a lot of controls for all kinds of possible issues. So what are the possible pathways? Well, same as for the stomach. So the what are called the core interoceptive pathways going through the vagus or the glossopharyngeal nerves, or through the spinal nerves. There are some additional pathways for cardiac interoception, which are quite interesting. So here, those which are related to the somatomotor system, where actually pulses of the blood in the vessels could be detected both by somatosensory receptors, but also actually by the muscles themselves. And more recently, there is now more and more evidence, including a very nice paper, which should be out in Science soon by Veronica Egger, showing that actually this can be transduced directly in the brain, where both glial cells and pyramidal cells, and in her case, mitral cells, fire in response to changes of blood pressure in the vessels. Okay, so this is something which is super interesting because it opens all kinds of new ways for the brain to sense heartbeats. So we embarked into a project where we looked at the expression of subjectivity across many different cognitive domains. and maybe I will begin with one where it's pretty obvious, which is self-related cognition. And here what we did was a simple interrupted thought paradigm, where it actually looks like a resting state experiment for participants where they fixate a dot for very long times. So it's pretty boring and they start mind wandering. And we interrupt them with a visual stimulus like this from time to time. And then we ask them to rate the engagement of the self in the thought they were having just before we interrupted them. So if you're thinking like, I don't know, how this talk is boring, then yourself is not much engaged in the thought. If you're thinking, I need to do the shopping this evening, then you're engaged as an agent in the thought because you are thinking about yourself doing something in the future. And you might also, of course, be thinking about yourself. I don't know, my back is aching, this chair is not comfortable. So we trained participants to do this kind of rating, and we looked at how the brain is responding to heartbeat during the thoughts they were rating, taking into account the ratings, whether they were highly engaged or not. And what we found is that in those two regions of the default network, we had larger responses to heartbeats when people were either engaged as an agent, so what is called the I, or when they were thinking about themselves in VMPFC, what is also called the me. And we were lucky enough to replicate those results in intracranial recordings in epileptic patients, thanks to a collaboration with Vincent Navarro at Hôpital Pitié-Salpêtrière. And we had one patient with an electrode in posterior cingulate cortex and another one with an electrode in VMPFC. And I'll just show you the results for this patient with an electrode in VMPFC, where you can see here on a single trial basis, the amplitude of the heartbeat evoked response on the x-axis and the self-rating on the me scale on the y-axis. And you can see this pretty tight correlation with how VMPFC is responding to heartbeat and what the patient is thinking about.
Catherine, can I interrupt with a quick question? So how do you exclude the possibility that the correlation you find between heartbeat and thinking about self is not that we're just more aroused, more interested when we think about things that are related to us directly?
Yeah, so that's a good point. So here we had as controls, we had something about whether the thought was emotionally positive or negative and whether it was more or less intense, and there was no correlation. Of course, we did not, so we also had something on time, whether it was, present, future, near future, near past, or far past. There was nothing interesting in there either. We did not have anything about whether it's more interesting or not, but I think it should have shown up a little bit in the intensity of the emotional ratings. Honestly, when we set up these experiments, we had a first step where we just collected the thoughts of participants just to have an idea. It was actually super boring in general. It was a lot about, this is boring, I'm cold, or I'm too warm, or what they were going to do just after the experiment. So I also want to show you one experiment where we show that heartbeat evoked responses are related to consciousness state. And here we worked in patients who are emerging from coma with disorders of consciousness. Okay, so they are out of the acute phase of coma. They have alternance of sleep stages and wakefulness, but it's very difficult to tell whether they're conscious or not. And it's one of these clinical diagnoses which is extremely difficult to do. So for those of you more familiar with it, that's a distinction between minimally conscious state and unresponsive wakefulness state. And we worked with a group of Stephen Lorez in Liège, and in particular, and we used their clinical diagnosis, which is based on two aspects. One, which is signs of intentional behavior. So if the patient from time to time is able, to follow a command or to show some intentional behavior, then it would be classified as minimally conscious. But then it's known that this type of classification misses a lot. And so they are also using a brain measure based on glucose metabolisms measured with positron emitting tomography, such that if the glucose metabolism looks almost normal, they would consider that the patient is also minimally conscious. So we use this clinical diagnosis to train a classifier where this time we use the EEG activity that was recorded actually at the moment of the PET scan. So we used heartbeat evoked responses, so short segments of the EEG after a heartbeat, to reproduce the clinical diagnosis. And then we used this classifier on data that were test data. So from other patients, we made predictions, and we tested whether those predictions were in accordance with the clinical diagnosis. And we had an 87% accuracy, which is not bad. But the important point was if we did exactly the same thing, but this time we take random EEG segments. So same number, same duration, but this time they are not positioned they don't have any particular relationship to heartbeats, then we can always classify it, which doesn't mean that there is nothing in general in the EEG that says something about consciousness, but the classification accuracy was always lower than when we take the heartbeat evoked response. So this shows that responses to heartbeats do convey some specific information on consciousness state. And then I will just briefly mention some other experiments. So one is on subjective percept, so testing directly this idea that responses to heartbeats interact with how we see things in this particular case. So here it was an experiment on a vision at threshold, just presenting this kind of grading, but at very low contrast. So participants miss half of them. And of course, with some catch trials where nothing is presented. And when we look at what's going on before stimulus onset, we find that the heartbeat evoked response, again, this time in regions of the default network, do actually predict whether you're going to see or not the stimulus. And interestingly, we could use sensitivity and decision criterion to show that in this particular case, heartbeat evoked responses behave as sensory evidence. So it means that the more responses to heartbeats, the more likely you are to see the stimulus. Of course, this is not visual information. This is in the default network. This is in response to heartbeats. And the way we interpret that is that when participants report, I've seen the stimulus, there is a part which is related to the actual visual system, the stimulus, but there is also a part related to the eye, to the subject who is perceiving, and that ultimately, to report I've seen the stimulus, it's a combination of visual information with eye-related information, and which is why heartbeat evoked responses behave as sensory evidence in this particular case. We also investigated preferences with this idea that if interrogating internal state is a default routine, it should particularly be present when you have to decide between options. But if, as we assume, it's a default routine upon which all forms of cognition have evolved, it should be present when you have to decide which movie you prefer. So cultural goods which have nothing to do with your physiological needs. So we actually had participants to rate their preferences for about 400 movies, and those are just two examples. And what we found is that if we look at heartbeat evoked responses in ventromedial prefrontal cortex and at the valuation signal in ventromedial prefrontal cortex as well, the larger the interaction between those two, the more reliable and stable over time are the preferences. So as if reading your heartbeat and integrating your heartbeat into the decision made your preferences for cultural goods more stable over time.
Catherine, just another quick question. It's fascinating stuff. Do you think it will also work the other way around? Let's say I can increase your heart rate in some way and then ask you to choose to rate preference for certain things. Do you think your heart rate can affect your ongoing preferences?
Yeah, so that's a good question. And thanks for the question, because it's something I forgot to mention. So in all those experiments, we also we measure, of course, the heart rate. So what we do is that we measure the heart rate precisely at the moment where we are analyzing heartbeat evoked responses. And we show that whatever we see in the heartbeat evoked responses is not related to a change in heart rate at that moment, which doesn't, so in those experiments so far, it's not a matter about heart rate itself. Okay, it's a matter of how the brain is responding to heartbeats, which doesn't mean that if we were to change the heart rate, we would not see changes. I don't know. But in those experiments, we measured it, but we were kind of hoping that we would not have on top of that a complication due to the heart rate.
Thanks.
And then I will finally show you one experiment on cell-father distinction in emotions, because of course emotions are, something I was quite reluctant to go into because it's first super complicated, but also because it involves peripheral changes, and this is well documented. And so here, what we did was actually, we show that heartbeat evoked responses can distinguish one's own emotion from others' emotion. And so what we did was to present emotional images. Sorry, I forgot to put on examples, but let's say you see the nice friendly face of Tane, who did those experiments. And then you have to decide when you view this, we tell you in advance, okay, you will have to rate your own emotion when seeing this face, or you will have to rate the emotion displayed by Tane. That would be the other condition. Okay, so during image viewing, we find the regular usual stuff, such that, for instance, the activity of facial muscles explains part of how you rate the valence of the emotion, whether it's your own emotion or someone else's emotion. But what we found interestingly is that the amplitude of the heartbeat evoked response, even before presenting the emotional image, actually contributes to your valence rating, but only for your own emotions. Which again suggests that when you have to judge, when you know you're going to have to judge your own emotions, then you start reading your own state more closely. Okay, so altogether, this suggests that we do have experimental evidence in favor of a link between heartbeat evoked responses and subjectivity across quite a range of different paradigms and domains of cognitive neuroscience. As I've said right from the beginning, this is correlational evidence, and we don't know if the mechanisms that I alluded to at the beginning are involved. We just have no clue, and essentially the mechanism of interaction between the heartbeat evoked responses and either external stimuli or cognitive activity are unknown at the moment. So we started with this hypothesis in mind that signals from the viscera could explain some of the properties of subjective experience, but we are not there yet. Let's say that we have enough evidence to think that it warrants further investigation and more mechanistic ones. One elephant in the room is arousal, which is related also to this question, why do we do anything at all? Because arousal is viewed as a brain-body state, as a kind of motivational force organizing behavior. And so we've been, of course, measuring arousal in different experiments. In the different experiments, I'm not showing you all the controls, but I'm still very puzzled by what is arousal, or whether there is such thing as arousal. And we really wanted to understand better what is arousal, or how many varieties of arousal are there, and do different varieties of arousal share some common neural substrate. But there is a big but, which is at the moment, if we want to review the literature, there are almost 50,000 papers in PubMed on arousal. And this is where Magdalena Saba came with this great idea. Then, well, let's use tools of natural language processing to do a semi-automated analysis of those 50,000 papers. So essentially, she extracted all those papers. She extracted the terms which are present in the papers, for instance, heart rate or reaction time or emotional stimuli, etc. And then she measured the term co-occurrence. So for instance, does reaction time co-occur very often with blood pressure, for instance, or does stress disorder co-occur with blood pressure? And then she used embedding and clustering to show that, well, reaction time does sometimes co-occur with blood pressure, but less often than with other stuff. So blood pressure co-occurs with stress disorder more than with reaction time, for instance. And so she generated clusters where dots which are close together are actually terms that co-occur a lot in the literature. And this analysis allowed us to find that there are seven varieties of arousal, or at least 7 domains in cognitive neuroscience talking about arousals. So emotion, of course, physiology, stress disorders, sexual behavior, sleep disorders, sleep, and cognition. And these type of clusters are very stable across a range of parameters of semantic analysis. And then in the next step, we ran meta-analysis of brain imaging studies per semantic group. And so, for instance, we took all the cognitive studies talking about arousal and ran a meta-analysis on that. All the emotion studies talking about arousal and ran a meta-analysis on that, etc. And so we do find networks for each of them, except for the sleep disorders, where there is no significant cluster, but there are also very few studies. And when we look at all those results, what we see is that there is one brain region, which you can see here in the overlap between those studies, that it consistently activated across all arousal varieties, except for the stress disorders, as well as for the sleep disorders. If we look at unthresholded results, it's also true, and which is in the right anterior insula. And that's the only place where we get something which is extremely robust against all kinds of parameters, also specific to arousal, so that if we take any other group of studies that do not talk about arousal, we never find such an overlap. And this is in a brain region, which is this granular insular area 7, according to the Jülich Human Atlas, which is not well known and for which it's not clear whether there is any animal homologue, actually. What is known is that this is a region which is related to surprise. Okay, so this is still ongoing work, at least, starting to write things up. So why do we do anything at all? Okay, unfortunately, I still don't have the answer to that question, but I hope I've convinced you that it's worth looking at interoception in the sense that it does explain part of spontaneous brain dynamics. It does show interactions with cognition, and in particular, in our experiments, it could be related to subjectivity. As I've said, we are not proving definitely that it's related to subjectivity. And there are also some experiments in the literature suggesting that you can also consciously pay attention or not to your interoceptive signals, and then this becomes a completely different story. Yeah, I think this is it. I just want to thank the clever people who worked with me on those rather new ideas, which mean that we were really brave, because when we began, it was not clear this was leading anywhere. And thank you all for listening to me.
Thank you very much, Catherine. Really fascinating stuff. So the floor is open for questions. You can unmute yourself and ask whatever you'd like. I'm sure that Catherine will be happy to answer. I can maybe start with a quick question before people are setting up. So if you find this beautiful relationship between heart rate and consciousness, all right, the conscious experience, and if we agree that consciousness is a continuum and you can be more or less conscious, so there's some different levels, would you say that when my heart rate is higher, like after a run, let's say, or of course when I'm nervous or excited, but also when I do something physical, My consciousness is richer?
So, so again, I want to emphasize that what we find is not differences in heart rate. So for instance, if we consider the patients we looked at in the consciousness state study, heart rate is not different between minimally conscious patients and completely unresponsive patients. So it's about how the brain is responding to heartbeats. This said, obviously, if you would be exercising, it would probably change the response to heartbeats, and this might alter consciousness. This we don't know. There are a couple of experiments on exercise training run by Sarah Garfinkel in the UK, but not really, she's not measuring consciousness state. But I think your question also relates to this question about arousal. So to what extent is also bodily state important? So here we try to have a bodily state which is kind of neutral in the sense that it's participants sitting in a dark room, acoustically shielded, relaxing, etc. So they are not sleeping, at least we hope, at least not at the beginning of the experiments, but they are also not very aroused. For whether changing bodily state would change consciousness state, I don't know. From our data, not necessarily massively. The tricky thing is that here we are more talking about a subjective experience in particular, and you seem to be talking about a consciousness state that might be more related to the difference between being asleep and being awake, with gradations within the wake state, which are then potentially related to arousal, which is still something that I don't know, I can't really figure out what it is and whether that's whether we really have two different dimensions, one which is arousal and the other one which is the content of consciousness. I'm not sure that's how it works, but it's a very tricky question, actually.
Yeah, and also, I mean, if you add to the mix what you can always add, which is attention, right? I mean, if you think about all these experiences where you just notice more about your environment, either that for your survival or for your enjoyment or mindfulness or whatever, uh, there's more or less that you can absorb. And yes, anyway, yes, I agree with you. It's very complex, but you illuminate a lot of it. Thank you. Questions from here. Ilana has a question.
Hi, thank you so much.
Hi, Elena. Nice to see you again.
You too. And this is such fascinating work, and it's really like broadening my thinking. And I agree that in general, like we should be considering body more in thinking about the brain. I think two questions. One is following kind of the same line you were discussing with Moshe, which is, and I know there's still a lot more to explore in this mechanism, but if the critical point that you're seeing is that the brain is integrating heart rate, or not heart rate, just the existence of a heartbeat, right, to its computations. What is it reading? I mean, if especially if the heart rate isn't actually changing, right? What's the readout that is helping it decide, this is what I'm feeling right now, or this is right? How is it helping the brain? How does it? Do you have a hypothesis how it interacts with other stuff?
Oh, thanks. Yeah. That's a super important question. We have actually tons of hypotheses. The tricky thing will be to try to formalize them and to test them appropriately. But maybe I can give a hint. So you could imagine something where actually the heartbeat acts as a resetter for ongoing activity. Okay, so you have a mechanism which clocks different regions of the brain and puts a reset onto ongoing activity. It could be a way through which it's not really signaling any bodily parameters per se, although I'm sure some regions do this signaling, but let's say that in ventromedial prefrontal cortex, I'm not sure. And that this is what is important, that there is a resetting of the clock. It could be or same thing for the stomach. It could be that it gives you an idea of the clocking of a particular region with respect to other sensory regions.
Mm-hmm.
So that's one possibility.
Yes, maybe in a sense, bringing together a lot of systems to just, increase their synchrony and then check in, right?
Yeah.
Do you also think about like the heart as one example, and you also talked about the gut, but there's also, arousal changes, lots of other things that do actually, are informative in terms of your emotional state or state of, could the heart be just one of the things that you're tapping in, but one that has a robust signal for you to, be able to pick up in your neural metrics?
Yeah, I think that's a very, I think that's also why I went for emotions last, because that's where it's the most tricky part. I think emotions and physical exercise are the two aspects which are very tricky for potentially different reasons, but they are difficult to conceive. And this all standing questions of whether you are actually your emotional feeling is related to your bodily changes, or it's the other way around. It's because you are feeling this particular emotion that there are those bodily changes. That's a never-ending controversy. I'm not sure I can solve that with the kind of experiment we're doing, but that's also why in the experiments where we looked at emotions, we actually looked at the response to heartbeats before any emotional stimulus is presented, so before emotional elicitation. But the fact that when to evaluate your emotional state, that you are interrogating your internal state is, I think, something that we would all agree on. I think what we are bringing on the table is that we are actually interrogating internal state at all times, not only in emotional context, which is also why I tried to use mostly non-emotional situations. And then, as we did, you come to this question about arousal, which is a very tricky thing, because actually it seems like a simple notion, but if you look at it from the body perspective, it's not at all, in the sense that for different organs, levels of arousal would not be the same. Like for the digestive system, actually arousal is super high. Like when the digestive system is super active, when we say that there is low arousal, that's a typical rest and digest. That's where the gastrointestinal system is most active. So the very definition of arousal as an entity is, I think, tricky. And that's why we tried to get into this with this semantic analysis of the literature, because it's not easy to pinpoint what's common to what we as scientists have in mind when we talk about arousal.
I have lots of more questions, but I think others do as well.
Can I ask a question?
Sure, Adam, go ahead.
Okay, so firstly, thanks for the very interesting and stimulating talk. Two questions. Firstly, the gastro signal, the EGG, is that always present, or is that only present after a person eats? That's the first question. And then the second question is, you mentioned on one of the slides that these interoceptive signals can be used for some sort of coordinate transformation between different sensory systems. And I find that puzzling. I mean, that's a very highly computational process that probably requires a lot of cognition. So how would a kind of a low level signal, if I were to just call it that, how would that help with the computational transformation between sensory coordinates? Okay.
So first question about the EGG. So yes, maybe I can put back the slides on the EGG. The electrogastrogram, so these signals originating from the interstitial cells of Cajal, it's generated at all times. And it's generated by those cells even when they are cultivated in vitro. So it's really a pacemaker property. Okay? What happens during digestion is that actually the amplitude of the rhythm increases up to the point where it entrains the contractions of the muscles. In our experiment, we always record people during mild fasting, so after 2-3 hours after a meal, because we are not really interested into digestion per se. So, yeah. And then the second question about how interoceptive signals could be used for coordinate transformation. Well, as I've said, it's not something I would be able to prove at the moment, but imagine that you have, if we want to keep it simple, imagine that you have a representation of the heart, of the stomach in one brain region, and of the heart and the stomach in another brain region, okay, in skin coordinates or in gaze coordinates. Then it means that you have anchor points from which you can deduce the rotation matrix. That's just one possibility, which is mathematically plausible. I'm not sure it's biologically plausible, but yeah.
But do you think that would be the case also for transformations between other sensory systems that aren't necessarily related to the heart, like visual auditory, for example, or something?
Yeah, that's why, I mean, that's why here I'm using the example of the auditory, the visual system. So if I go back to the results, so we do find heartbeat evoked response in the visual cortex, and we do find coupling with the gastric rhythm. So let me just show you again, the gastric rhythm is massively represented in the visual system. And we do also find heartbeat evoked responses in visual regions. Like in this experiment here, we do find them in the ventral and dorsal occipital cortex. So again, that was not necessarily expected to begin with. In the first experiments, we had heartbeat evoked responses mostly in the default network. But we do also find them sometimes in sensory regions. I think, as I've said, those are kind of hypotheses that are driving our experimental work, but I'm certainly not saying that we are proving that those hypotheses are correct. I think what we're showing is that there is more than one would have expected from classical textbooks about bodily neural responses to bodily signals in cognitive tasks. That would be the lowest conclusion. Yeah.
Thank you.
Okay, maybe time for one more question.
So I'll ask a question. Oh, no, go ahead.
Yeah, I think let over.
Thank you. There is any knowledge or assumption about what the hairs represent at the level of the biochemical activity in the brain?
Sorry, I'm not sure I heard completely. It's about chemicals, but I missed the beginning of the question. I could not hear properly. I'm sorry. There was
Okay, I will talk now. I'm more audible. You can hear me now?
Yeah.
So is there any knowledge or assumption about what the heartbeat evoked responses represent from the biochemical level activity in the brain?
Okay, so thanks. That's a very good point. No, very little is known. As I've mentioned already, little is known about the pathways. There are those classical pathways, there are those newly discovered pathways, there are somatomotor pathways. And then about neurotransmitters not known, whether some neuromodulatory systems are more involved than others. To the best of my knowledge, it's not known either. This is something very frustrating about all this, is that I'm coming from the visual system where we know a lot, and this is typically systems where we know so little.
Thank you.
Go ahead, Ilana. We don't want to suppress your question.
Oh, it's fine. You can continue offline. I don't want to take everyone's time.
Okay, thank you very much, Catherine. It was really fascinating stuff. We look forward to to seeing more of your results in the future. Thanks again.
Thanks again for the
Thanks, everybody.
invitation.
Thanks.
Thank you.
Bye.